Now, let's talk the different types of antibodies. Hopefully you remember from previous chapter, we talked about that there are multiple classes or isotypes of immunoglobulins: IgA, IgD, IgE, IgG, IgM. Now, what we have found is that the B cells can switch between the two classes. Starts with one, but through different signals is told to come up with something higher affinity and for a different use. So as we're going to see in later chapters, the different immunoglobulin classes have different uses because of their structure. The way we distinguish between these 5 classes, the isotypes, is by their heavy chain regions. The variable domains, light variable, heavy variable, relatively the same through all isotypes. It's the constant regions are going to be different. The C regions, the constant regions, will offer support for the variable regions. And as we're going to see over the next few slides, the different constant regions are going to have slightly different domain structures. This is what these different domain structures are what are going to give the different antibodies their function. IgG, serum. IgE, across the mucosa. IgA goes in breast milk. IgM, and we're going to see, is the first one expressed, and so on. They each have their own role in the immune response. Have different roles as we saw here. Well, we're going to have some genetic recombination. Mature, naive B cells. So these are matured. They have left the bone marrow, circulated through the blood, are now in a lymph node, waiting. They express high levels of IgM and to a certain degree IgD. Has not been activated, matured, activated to become a plasma cell. So it hasn't gone through any class switching as of yet. Once activated, and it's going to be activated via some IgM, IgD on the surface acting as receptors, you will then see the VDJ will remain as is on the heavy, but we're going to get swapping of either the CM or the C delta to either an A alpha, or the A, the E, or the G. Now, if you want a transmembrane secreted, basically you want this antibody to act as the receptor for this B cell, then what you have to have is what is referred to as the MCP on domains. What this will allow is a transmembrane tail to be added during RNA processing. All of this will be added, and then during translation you will get a hydrophobic tail that will insert into the plasma membrane, whether it's M or the IgD. Now, during RNA processing, if this portion here is cleaved, then you get just the VDJ binding to the C constant M segments, and a poly A tail, you get what is referred to as a secreted immunoglobulin. The secreted immunoglobulin will now go into the bloodstream. It will be excreted out of the cell to enter into the tissues or interstitial tissues or into the bloodstream itself. It's also in this portion that we find that the sequences will allow for joining of certain antibodies. So they can either be a secreted tail or a tail on the antibody that will cause it to be membrane inserted, loss of that tail for secreted, or a tail that will allow for specific joining via what they call a J-chain protein. The J-chain joins, here showing IgA, joins 2, and then disulfide bonds join each of these immunoglobulins to the J-chain and then join the immunoglobulins to themselves. So primarily, you're going to see most IgA is in this dimer form. IgM, for the most part, is going to be formed into these pentameric, one J-chain, and then numerous disulfide bonds joining all 5. Can you find singular IgA? Can you find singular IgM? Yes, but the majority of it's going to be either a dimer or a pentamer. Now, keep in mind, this J-chain has absolutely nothing to do with the J region of the VDJ of the heavy chain or just the VJ of the light chain. Okay, here J-chain means just joining chain. It's a joining protein. It's not the J region joining of regions. What's interesting is because this is the first antibody that is generally going to be made by a B cell, it's the first try. It's the rough draft. Very little recombination. Very little hypermutation has occurred yet, so the IgM has been shown to have the lowest affinity or the lowest binding potential to any antigens. During the recombination events to go from M to D or M to G, M to A, M to E, we also see the recombination occurring. We see the hypermutation and a significant increase in affinity. So, class switching, and now we see we can expand our repertoire of antibodies by going from M to G, M to E, whatever. But then we also take into account things such as somatic hypermutation, which V to which J, which VJ to which C, and so on. And then we got to take into account gene conversion. Gene conversion is going to allow for the swapping of, after the class switch, how do we get, increased affinity? All of these are going to be using an enzyme known as activation-induced cytidine deaminase, or AID. Notice, all 3 initiated by the enzyme. Activation-induced. This means that mature, naive B cell, once it gets induced, it's now going to go from naive to a plasma cell. It's going to start making more antibodies. So it has to be activated. No activation, no AID, then that B cell is going to stay at IgM and will not switch classes, will not hypermutate, and none of this. And over time, that B cell will hit its shelf life, and potentially will just die off, taking its gene rearrangements with it. It means you are never going to be exposed, or never will be, never have been exposed to that antigen. So when you look at it, this is our first antibody B-cell receptor for that naive, mature naive. Gets activated, you're going to get some somatic hypermutations, which is going to change regions in the VDJ for the heavy and just VJ for the light chain. Class switching is going to allow for M to G, M to A, M to E. And then the gene conversion, as we see here. We're going to take V and we're going to swap it around for one of our V pseudogenes, or a different, we're going to recombine it with, V1 segment with part of V14. See if this makes it just a little bit better. Keep in mind, after this initial B cell has been activated and it's becoming a plasma cell, it is also going through clonal expansion. So everything we see here is not happening in just one cell. This one cell becomes 4. As these 4 are maturing, they're also splitting, becoming 2 or 3, 4 themselves. Some of them are just doing a class switch. Some of them are doing hypermutation and class switches. Some are doing class switch and gene conversion. So you get this randomness involved, so that whichever plasma cell gets the highest affinity is the one that's going to continue to get the most signal to keep producing. Now, how this all works, remember, this is AIDE, our activation-induced enzyme. What this does, AIDE goes in and deaminates cytidine. Turns cytidine into uridine. So what you end up with is a mismatch repair situation. So instead of being G to C, you are now G to bastardized uridine, which is not a DNA nucleotide. So what you end up with is something that is quote-unquote normal in DNA, and now you get something that is uracil-like, but not really, and should only be an RNA, maybe. So you're going to have normal mismatch repair enzymes coming in to fix. But what's going to happen? Well, AIDE comes in, turns cysteine to uridine. You get mismatch repair. You end up with mutations. Maybe what was CG is now AT, and you get a slightly different amino acid, somatic hypermutation, potential change secondary, tertiary, quaternary structure. AIDE switch cysteine to uridine. You get base excision, uridine to abesic, Rev1, mutations, CG. Now it's flipped. What was CG is now GC, somatic hypermutation, potential. AIDE, cysteine, uridine, uridine to abesic, cut it out. Boom. Gene conversion. You get a single strand break that you're going to loop around, and you're going to get some conversion recombination. Or it goes through and does a complete double strand break. If you have two cysteines that were close, cytosines that were close to each other, in the DNA sequence, you're going to end up with the double-strand break, and then you can end up with class switching as a means of repairing the damage. Hypermutation gives us a lot of rearrangement in the immunoglobulin V region, the V domain. You look at early on, so we're looking at after the primary response. So we're going from mature naive to a mature activated. Very little antibody made. Most of the originally here are going to be M. And then we start to see as it goes and does the class switching M to something else and all those others, the mutations in the variable regions. CDR1, CDR2, CDR3 are hypermutation regions. Begins to accumulate week 1, week 2, week 3 of our antibody response. What this is showing as our one initial B cell starts going through clonal expansion. 1, 2, 4, 8, 16, 32. Now you've got hundreds, and within those hundreds, you start to see the accumulation of more and more point mutations. The point mutation, good, bad, neutral. If it's bad, that cell dies off, doesn't get simulated, but the ones that it's good or neutral keep going, and we see the accumulation. So that by the time we're 3 weeks after that B cell has been activated, what we are now expressing as an antibody in no way, shape, its variable or hypervariable regions in no way resemble what we started off with.
Naive B cells can only express surface immunoglobulin of the IgM and IgD isotypes. In these cells, the heavy chain immunoglobulin locus has previously undergone VDJ recombination that placed rearranged V, D, and J segments upstream of the C-mu and C-delta constant regions. Transcription initiated upstream of the rearranged VDJ segments can produce alternatively spliced mRNA transcripts that generate either IgM heavy chain or IgD heavy chain proteins. After translation, the functional IgM and IgD proteins associate with light chain proteins and become expressed on the cell surface. In this initial orientation, transcription of immunoglobulin arises only from the promoter located upstream of the rearranged VDJ and produces only IgM and IgD isotypes. The gene segments encoding the constant region for all of the remaining isotypes are located downstream of the C-mu and C-delta constant regions. In the mouse genome, these constant regions occur in the order of C-gamma3, C-gamma1, C-gamma2b, C-gamma2a, C-epsilon, and C-alpha. With the exception of C-delta, each of these constant regions is preceded upstream by a sequence of DNA known as a switch region. Switch regions range in size from a few kilobases to nearly 10 kilobases in length and are composed of hundreds of repeats of G-rich short DNA sequences characteristic of these regions. For example, the switch region located upstream of the C-mu constant region is approximately 3 kilobases in length and is composed of approximately 150 copies of this short 21 nucleotide element, which itself contains 3 copies of the pentameric sequence GAGCT. Located upstream of each switch region is a DNA sequence that functions as a promoter, called an I region promoter. Under appropriate circumstances, the I region promoter may be induced to initiate transcription by RNA polymerase II. Transcription begins just upstream of the switch region and continues through the region of DNA encoding the constant region. When this first occurs, the messenger RNA produced is known as a sterile transcript because it lacks an initiating methionine codon and therefore cannot be translated into functional protein by the ribosomal machinery. Isotype switching is stimulated in the course of an immune response. An activated T follicular helper cell engages a B cell through linked recognition. The follicular T cell expresses CD40 ligand and produces cytokines. In this example, interferon gamma. CD40 ligand engages a CD40 molecule on B cells that activates NF-kappa B. And interferon gamma activates STAT1 within the B cell. These transcription factors cooperate to selectively induce the transcription of the I region promoter upstream of the C-gamma-2A constant region. However, they do not induce transcription from other I region promoters, such as C epsilon or C alpha. The different transcription factors activated by the cytokines determine the specificity of I region activation. In this case, activation of STAT1 by interferon gamma leads to the activation of the I promoter region for C-gamma-2A. Induction of transcription in the I region promoter opens up the DNA and allows an enzyme known as activation-induced cytidine deaminase, or AID, to access the switch regions. As RNA polymerase II transcribes the sterile transcript, the repetitive elements in the switch region induce a behavior in the polymerase that allows AID to deaminate single-stranded cytidine residues in the DNA and convert them randomly to uracils throughout the switch region. Through the action of two other enzymes, uracil DNA glycosylase, or UNG, and apyrimidinic endonuclease, or APE1, these uracils are eventually converted into single-stranded nicks in the DNA on both of its strands. These processes also occur at the C-mu region, or any other switch regions actively transcribed in the B cell. These activities create multiple breaks in the DNA of the mu switch region, as well as the DNA of the switch region selectively activated by cytokines. Because the breaks have been produced in two distinct locations at a great distance from one another, the double-strand break repair machinery joins the two switch regions and excises all of the DNA located between the two damaged regions. After the double-stranded DNA break repair machinery finishes its work, the damaged switch regions will have been spliced together at some random point, leaving remnants of each switch region on either side of the repair joint. The rearranged VDJ segment is now placed a few kilobases upstream of a new constant region, in this case C-gamma-2A. This now makes it possible for transcription initiated from the V region promoter to generate a transcript encoding the original variable region, but now in the context of a different constant region. When the messenger RNA is fully spliced, the intervening switch region sequences are removed, leaving only the segments encoding a fully functional C-gamma-2A heavy chain. The B cell that has undergone isotype switching will begin to secrete antibody of the new isotype class.
So, as we saw in that video, isotype switching is going to be a direct induction by a CD4 T helper cell. CD4 T helper cell and the signals that are passed on from it. In the video it showed interferon gamma. What we see happens is that this is a functioning VDJ segment, okay? Because the B cell receptor was able to get an activation signal, okay? The receptor and its co-receptors there were able to function to get enough of a signal to activate the B cell. The CD4 T helper cell is now going to induce the switching from IgM to a different class. So we want to maintain the VDJ, but we want to switch from M or D to G, A, or E. Keep in mind, the VDJ that was expressed, the variable heavy regions, this was determined in the bone marrow. So the cells, the signals that were nurturing this immature B cell, this developing B cell in the bone marrow. They are the ones that help determine which V to which D to which J, beyond just the randomness. So it was determined that the VDJ works there. That cell was released to the bloodstream as a mature naive. It's now being activated, and it's getting signals from CD4 T helper cell. Time to switch classes. The only thing we see here, the only way that the V region can be changed here is by hypermutation, point mutations that we've discussed before. APE1, UNG, AID, specifically AID, these are going to be a little bit more B cell oriented, a little bit more isotype switching oriented. Double strand break, well, this is just a generic repair enzyme complex. This generic repair enzyme complex is going to come and bind up this region. Another complex is going to bind that region, and it's going to bring them close together and excise out everything in between. Join together, and this S region, this signal region here, where we had the promoters for mu, for epsilon, is no longer anything of want, so that during transcription, promoter up here, transcribe through VDJ, transcribe this as one long intron, and then transcribe C epsilon.